Welcome back. I hope you enjoyed lunch and had some good discussions. One more talk in this bio-inspired paradigms grouping. So we've got Caswell from Imperial who's going to tell us about what AI can contribute to neuroscience.
Thank you very much. I'm from UCL though, not Imperial.
Sorry.
But, interchangeable London universities.
No, it's not. But I should know that. I was reading across the lines wrong here. My apologies.
Don't worry, don't mind. Okay, so thanks for inviting me. I'm a neuroscientist, so maybe I'm speaking to a different audience of people than I'm usually used to speaking to, and I'm maybe used to dealing with slightly different problems that you are. But hopefully there's some common ground, as we'll see. And so what I'm going to contend today is that machine learning, AI, as it's popularly called these days, has something useful to contribute to neuroscience above and beyond what it's just going to contribute to the world in general. And so hopefully by the end of this, you might believe that statement. So what is neuroscience? It may sound like a trivial point, but it's not, right? So it encompasses a huge range of sort of techniques, everything from people working on individual ion channels, trying to understand how an individual neuron integrates information, up to, I guess, labs using things like fMRI and looking at patients, trying to understand how whole networks work. I sort of sit in the middle of that, in that my lab typically tries to understand how networks of neurons in specific areas of brains, normally rodent brains in our case, works. Most of what I'm saying is more applicable to people who are interested in something neurons, plural sort of level, not individual neurons. And so what are we trying to do? Essentially, we're trying to understand how the biology generates cognitive phenomena. So how can a bunch of neurons mean that I remember or can find my way around, for example? And there's specific questions within that we're trying to tackle. So for instance, even if I have access to the information in the brain, I might try and understand what information is present at a particular place, particular level of neurons. How is it encoded? How are stimuli from the outside world represented in the brain? And what functions are being performed by different circuits within the brain? So how is information transformed as it travels through from the sensory surface, ultimately to affect the behavior of an animal or human? And really we've got two problems. This is very generally speaking, but there are basically two problems. One of this, the data problem, we're very good at generating large amounts of data. We have techniques such as fMRI will typically yield a couple of gigabytes of data of brain, say half an hour. Electrodes with a few hundred electrodes, which is commonplace these days, recording from a brain, I can generate about a gigabyte of data a minute. Things like 2-photon microscopy, which lets you image the sort of neural activity of hundreds, if not thousands of neurons at the same time, is somewhere above that, and it's increasing all the time. So it's not the quantity of data that's the problem, it's understanding what's actually present. Like if I've recorded from a neuron and I see a series of action potentials, those are the bursts of electricity that tell us it's active, can I infer what's actually being encoded in here? Indeed, do I even know what the coding scheme is? We know quite a few things already. For example, we know the frequency is important, but we also know the timing relative to what other neurons the network is doing important. We know that changes across different brain areas, so it's not a trivial question. That's the data problem. The other problem is at the other end of things, that essentially there's a bottleneck in understanding, in building models of how does this all fit together. If we want to claim to understand the brain at some point in the future, we're going to need to build models. And models is just, in a sense, another way of saying hypothesis. How do I think this works? Model is just a complicated hypothesis that I can write down and share with other people. And we're the bottleneck in that at the moment. I could collect some data, sit around, wait for inspiration to strike, and then say, hey, I think these cells that I've just recorded work like this. And then we could go on and have discussions about it and argue about it. You might prove me wrong. But I'm fundamentally the bottleneck there. And we want to improve both of these areas. And I think the important thing I'm going to say today is I think AI has got something to add to both of these fields. What do I mean by AI? AI is obviously a much maligned term. It's used in many different ways. It seems to mean everything from a GLM all the way up to the deepest deep network you could possibly think of. What I'm thinking is that, in particular, deep networks have got something special to say about neuroscience. And there's a sort of obvious trivial version for that, which is this. That they are fundamentally inspired by things we know about the brain. They're very abstracted versions. There's certain things they don't do or don't commonly do, but certainly they're inspired by it. There's common architectures. There's nodes, which are analogous to neurons. There's synaptic weights, which are analogous to the the weights in a biological network. And so computer science has been very good at doing this, taking inspiration from the brain. So the fundamental unit of a neural network is modeled on neurons. Things like convolutional networks are modeled on CAP visual system. Things like recurrent networks, fundamentally like the hippocampus, which is highly recurrent in the brain. Things like deep reinforcement learning, informed by behaviorism, which we know that animals use as a way of updating their beliefs about the world. So computer science has been good about doing things like this. What we need to do is turn this around and take inspiration from deep learning and try and apply it to our understanding of how the brain functions. And so I think there's two ways really this can happen. So we can tackle the bottom-up problem. Deep learning is very good at finding structure in noisy data. We know this because it performs well on things like the identification of images, visual, noisy images. We can train the network to perform well above human level ability now. So this is part of our problem, right? Finding what is present in neural data. After we've trained a network, we can go back and interrogate it and figure out how it's doing it and maybe get some insights about what is important in that neural data. But this is kind of true for many empirical sciences, right? If you want to understand complicated data you're recording about, I don't know, the environment or something, this would still be true. This isn't though. This is special to biological systems. So it seems plausible, and I'm going to show you why I think this is plausible, that deep networks can function as models of the brain. If we train a network to perform something the brain does well, then potentially the solution the network comes up with is going to tell us about the solution the brain came up with. And because we believe this, because of the commonalities, the common history these fields share. And so I'm going to demonstrate a couple of examples, one from this and one from this, hopefully convincing you of these things, and there's a future here to be had. So let's go down with the top down, the building a model approach first. So can we use a network as a model of the brain? So this is a project that my lab did in conjunction with DeepMind. So I was lucky enough mainly to work with these three guys here who have joint DeepMind UCL affiliations. I've only got a DeepMind affiliation, a UCL affiliation. Maybe I wished I had a DeepMind. And Andrea is my PhD student who did most of the work on this. So let me tell you something that animals can do well, and let's see if we can understand how this is implemented in the brain. So this is a famous, if not slightly esoteric looking apparatus from the 1940s. So this chap, Tolman, spotted something interesting about the way rats navigate through space. So he trained these rats, he put them in this central area, and he trained them to run down here and go and get some reward over there. So far, so simple. What he noticed is that if he blocked off this eventuality and then gave the animal a bunch of different choices, they would tend to shoot off over in this direction here. So that may sound fairly straightforward to you, but at the time it was very surprising. Stimulus response ideas held sway and people assumed complex behaviours were just broken down into simple things that could be learnt and chained together. So for instance, the animals just learnt to go forwards, turn left, turn right, etc. and get to there. They shouldn't have learnt anything about the structure of space, but the very fact the animals could do this tells us they'd learnt something extra about the way these different places related to each other. Interestingly, a slightly apocryphal story tells us that he noticed this because there was a cover on here and one of his rats climbed out onto the cover and ran across the state to get at the food. And so the question is, it appears animals have information about a spatial metric. That is, they know about the Euclidean distance and direction between places in space, even though they haven't potentially been through that exact route. So for instance, I could probably point to the toilets, which I think are over there. I've never directly travelled through that line, but yeah, I have information about that structure, the structure of space. And how is that encoded in the brain? How does the brain do that? How do we perform that calculation? Knowing that is useful for a neuroscientist, because we want to understand how the networks generate cognitive phenomena, but it's also interesting for people who want to build, say, agents or robots that navigate better. Luckily, we already know quite a lot about this. That work was done in the 1940s, and there's been essentially 50 years worth of research looking at what the brain does that might be useful. So one of the first things to be discovered in the 1970s was there's an area of the brain, the hippocampus, which is shared amongst all mammals. And if you record individual neurons from the hippocampus in an apparatus like this, you have a rat, you record from a neuron in the hippocampus, say this is a 1 meter square box. The animal might run around in that box, that's the black line, and a given neuron will fire in a given place. Doesn't depend on the direction the animal's facing, what it's doing, as far as you can tell what it's thinking, it's just representing a position in space, and they were called place cells. They were discovered by John O'Keefe, who got the Nobel Prize for them about 5 years ago. what was discovered much more recently in the 2000s was a sort of matching category of cells, which were termed grid cells. They have many of the qualities of place cells, and so I should say these things here are incredibly stable. I took the rat out of the box and put him back in a week later, assuming I could find the same cell, that cell would still fire in the same place. And so animals apparently have hundreds of thousands of these. There's enough information for us, and presumably the rest of the rat's brain, to accurately decode where the animal is in space. Place cells, however, are partnered by another type of cell, which were termed grid cells. So whereas this place cell fires in one place, this is an individual grid cell. So you've got the black line underneath, and each of these dots is a single dot every time an action potential is fired by that grid cell. And so you can see it fires in multiple locations, each of these nice little circular fields. If I turn this into a frequency map, you can see it slightly better. You can see these little fields are laid out in a nicely tessellated triangular structure. And so again, rats and indeed humans, we now know, have many of these things. And so what it looks like is this appears to be something that functions a bit like grid lines on a map, except they're triangular, not rectangular. And these are maybe indicating interesting features. And so it's also known that animals have grid cells of different scales. So this distance here, you could call it a scale. So we might say this one's got a 20 centimeter scale. But animals, we know, have multiple scales going up to several meters. And so potentially, well, let me give you this example first. This is what these things look like when they're recorded. So here's a rat. You'll see it running around in the box. And overlaid are individual dots that will start to appear about now, which are single action potentials coming from an individual grid cell. So the point is, there's nothing hexagonal about the world. There's nothing hexagonal about the animal's behavior. But yet slowly, it'll speed up in a second, this hexagonal structure will start to form. You can hear, by the way, that noise you hear is what we hear in the recording room. So that's the, you can't quite hear it, but we typically play back over a speaker the sound of individual neurons so we can hear them. Anyway, the exact details aren't too important, but as he moves around, you start to see the individual grid fields forming from an individual cell. And so there've been a number of sort of ideas and theories over the last few decades about how these sorts of cells might support navigation in the world. And indeed, because place cells were discovered first, nearly 50 years ago, and we know animals have lots of place cells, there are a lot of ideas about, well, maybe this is the basis of these sorts of calculations. If you have lots of place cells, presumably you could use them to make shortcuts and do things like this, take a shortcut to the toilet, get back to your escape hole if you're a rat. Actually, there's a problem. These individual fields, while they give you a lot of information about where you are right now, they don't tell you about how different places are necessarily related to each other, precisely because of this. If I stand here, there's a bunch of place cells firing that are unique to here. If I come over here, there's another bunch of place cells firing which are unique to here. And knowing this vector of place cells and this vector of place cells doesn't tell me anything about how those places are relative to each other. They could be a couple of meters apart, they could be in London and New York. It's no good for large-scale navigation. It'll let you get the local topography, but it's no good for large-scale navigation, which is precisely why people started to get excited by grid cells when they were discovered in 2005 and why these got the other half of the Nobel Prize. And so it turns out that because you have different scaled grid cells, you have smaller scales. We think animals have got about 8 different scales. It turns out it's very likely to be a combinatorial system that not only gives you very efficient information about where you are in the world, can also encode other places. And if you know the grid pattern for the other place and the grid pattern for where you are now, theoretically at least, you can infer the vector between those places and use it to take shortcuts. And so that's how the state of play existed before we tried to tackle it. Theoretically, grid cells are a great substrate for doing vector-based navigation. The problem is, it's really hard to test it in animals because we don't have the tools that can go in and tinker with the individual grid code. So we needed a system that allowed us to go and ask this question, are grid cells useful for vector-based navigation in a real system? And so what we tried to do was this. We have 2 aims. First of all, we wanted to see if a deep network could be trained on a biological-like task, whether, if it was trained that way, whether it would evolve representations that we see in the brain. Second, if we did that, if we evolve systems that we see in the brain, we wanted to understand whether we could use that as a tool for testing biological questions, whether we could do experiments on the network instead of experiments on the animals, practically. And so we chose to work on these sort of spatial navigation tasks, mainly because my lab does a lot of spatial navigation, but also because this is already a well-established, mature field with strong theories about what these different sorts of representations do. So we started by doing this. First of all, we wanted to see if we could develop grid cells in an artificial network. So we had a simple task, it's relatively simple, a network trained through supervised learning. So we said, you start somewhere in the world, we'll tell you what some place cells are doing there, because we know the brain has access to place cells. We'll tell you how you move, how you've translated, turned, and we want you to predict what place cells are active at the next step along. And so this is something that biologists would refer to as path integration. We know animals are very good at this. You can update your own representation of where you are based on how you're moving around. If you get up in the night and it's dark, you can walk to a light switch and turn on the lights, because you've got a sort of basic model of the world and you can integrate your own motion. And so we trained a network to do that. And so basically we have a simple feedforward network. There's an LSTM layer in there. LSTM just stands for long short-term memory. It's essentially a moderately complex form of recurrent network. That's filtered through a linear layer. And the linear layer is there purely so, well, for two reasons. One, because it's easier for us to interpret. And two, because it gives us an easy way of imposing something known as a dropout. So it's essentially a sort of noise that's roughly analogous to the noise that you see in a biological system. And we're asking the network to predict what place cells are active at the location it's moved to, as well as what head direction cells. So head direction cells are a sort of partner type of cell that you find in the brain. Place cells tell you where you are, head direction cells tell you which direction you're pointing in. So we train the network to do this through time. The first point is, it's very good at this. No one would be surprised if it wasn't. This is exactly the sort of thing networks are good at. If you tell it where it is to start with, you give it information, it can, the smooth blue line here is the actual direction of movement. At each time step, it's very good at tracking where it really is. That's not surprising. When it's not trained, it's not good at it. When it's trained, it's good at it. That's not the point here. The point is what's going on inside the network. And so now we went and inspected this linear layer to try and understand what sorts of representations we were seeing in there. And so what we found was this. These are maps showing you the average activity of individual nodes in that linear layer over the environment. So these, as you hopefully will see, look a lot like the grid cells I was telling you about. Maybe I should tell you a story. So my lab's made up of experimentalists and theoreticians, and so rather meanly, I showed these maps to one of the people doing experimental work who didn't know we were working on this. And their first reaction was surprise, because they didn't expect me to actually be doing any experiments, and also that these were very nice grid cells and I'd done well that day. So these are, I'm telling you this to tell you, these are biologically plausible. These are plausible in the sense that they would trick an experimenter who's used to looking at these day in, day out. So we saw these things that look like grid cells. We saw this thing here, which has already got a name in the biological literature. These are called boundary cells, so they tend to fire along the boundaries of animals' environments. We also saw this thing down here. This plot is just telling you whether the activity of the cells are modulated by head directions, the direction the agent's moving, as this would be a very typical head direction cell, which you see plenty of them in the entorhinal cortex, which is where you find grid cells. So, point number one, we found in this network something that looks very, very plausible. It looks like what you'd expect to find in a biological network, not only the grid cells, but also the other stuff. These accounted for a reasonable proportion of our units. So we had 512 units in the linear layer, about a quarter of them the grid cells, which out of interest, a few people say, well, why didn't you get 100%? Actually, in the brain, in the entorhinal cortex, you get about 20-25%, so that's pretty much odds on. We found the head direction cells. We also found quite a lot of unclassified things, which also isn't surprising. If you go and record from the entorhinal cortex, you find a lot of unclassified things we just don't understand what they're representing. And so that's not necessarily a bad thing. We also found that we had grid cells of differing scales. And so what I didn't tell you, so you can see this is a multimodal distribution. There's a preference for the scales to be clustered at different values. And indeed, there appeared to be a common ratio in between the grid scales in the network. You might think that's very odd and the sort of thing you wouldn't find in biology, except it is exactly what you find in the brain. So there's been an ongoing debate about exactly what the ratio is between grid cells in the brain. And the argument is currently sort of oscillating between whether it's somewhere between 1.4 and 1.6. So actually this lies pretty much in the middle of the range we see in the brain. And we also see cells that are conjunctions. So you see grid cells that are tuned by head direction, which is exactly what you find in the brain. So this is looking good so far. We've ticked off aim number one. This looks a lot like the brain, but can we use it to test things? So we wanted to know whether this network can now perform tests that animals are very good at. And so first of all, we tried something called a Morris water maze. Now, Morris water maze is if you're a behavioral neuroscientist, is a very, very famous task where you basically have a swimming pool about so big, you fill it full of milky water so it's opaque, and you have a platform that's just under the surface, and you put a rat in it. Rats can swim really well, but they don't like swimming, so they'll swim around, they'll find the platform, and if you put them back in again, they'll go straight to that platform. And it turns out to be a test that is incredibly sensitive to damage to the hippocampus, where you find place cells and grid cells. If you damage the hippocampus, you really can't find that platform again very well. So we came up with our own Morris-Watt maze. Open field, this is a dry version if you like, but it's done in virtual reality. Open field, there's a goal you've got to get to, it's not marked, there's cues in the background, so you can figure out where the goal is relative to the cues and how you're moving, but you need to remember it and you need to be able to perceive the space and navigate. And so our network, this is our network from the last step, so our network could actually interact with the world. We had to take the simple supervised network and put it into a much more complex reinforcement learning trained network. So guessing most of you know what reinforcement learning is, but reinforcement learning is just a way of training a network where there are where you get feedback every now and then about how you're doing. In this case, the aim of the network is to maximize the expected cumulative reward, which means get to that goal as many times as you can. Each time you get to the goal, you get dropped back into another location. Every approximately 10 trials, then the environment changes, the color changes, the goal location changes, so you can't just overlearn one strategy, you have to learn to navigate. And so the basic network is incorporated in a much larger reinforcement learning trained network which receives the grid cells. It also has convolutional networks so it can perceive the world around it. And then it has another recurrent network which controls its policy, what it's going to do. So we found this is very effective. First trial, the dotted line, it explores, finds the goal. Next time you drop it in at random, it can navigate very, very quickly to the goal. And it's really good. So here's how it performs over training. Here's the grid network, the one we're talking about. Here's sensible but powerful alternative network, so grid network where we've scrambled the grid cells, or just a general sort of state-of-the-art reinforcement trained network. They don't do half as well. Here's how a human did, and by the way, I should say this is no ordinary human. This is a professional games player. I tried this, and I'm down here somewhere, I think. So it's doing really well. It's nearly sort of saturating. It's nearly doing this as well as you possibly could, given the distances involved. But what we really want to know is whether it can do this sort of flexible. You can get to the goal. There's lots of ways you can get to the goal, but can it do this sort of shortcut behavior? Can it take a route that's never taken before because it knows about the structure of space? And so we tried to set up our own version of this famous sunburst. By the way, I should have said this is called the sunburst maze for obvious reasons. So we tried to set up our own version of this. So basically the agent starts on one side of here. There's some doors. Only one of the doors is open. It's the one on the far side. It has to get to the goal. Gets one shot at doing that. Then we drop it back in again and we open all the doors, and the idea is to see whether it sticks with the inefficient route-based strategy, which would mean you come around here and go that way, or whether it's able to utilize what it knows about the structure of the world and take the shortcuts. And obviously, as you can see here, I probably wouldn't be telling you this if it wasn't true, it's able to take those shortcuts very efficiently, and it prefers one of these two doors that is the direct line route. And so the agents that don't have grid cells aren't able to do this sort of thing. So that looks pretty convincing. And quickly, without going into too much detail, it also works in much more complex environments. So here's an environment that, again, changes on a regular basis. It has multiple doors that can open and close stochastically, so you can never be sure which route you're going to take. In environments like this, it also it shows an excellent level of performance. It exceeds the human, it exceeds the other control agents. And because we're neuroscience, we're specifically interested in whether it's using this vector-based navigation, we could go into the agent and say, can we actually see any evidence that there are these Euclidean quantities? Is it really calculating and representing the vector to the goal? And so indeed you can show that you can decode the vector-based quantities from this, whereas in other agents you can't. So yes, this looks like a tick. It looks like we have an agent that's got biological quantities and it's able to exhibit vector-based navigation, so we can use it for doing experiments. We've kind of demonstrated that grid cells are useful for vector-based navigation, which was the aim. Very quickly, because I've only got a minute or two left, I notice, I want to tell you about a project we're working on from the other end, so the bottom up. That's the top down. We've used deep learning to build a model of how we think the brain might work. Can we turn this round? Can we do something bottom up? If we've got some neural data, can we understand what's actually present in it? So let me tell you the problem. So I should say this work was done by Sandra and Marcus. Sandra records the data, Marcus trains the networks to kind of try and understand what's in the data. And so again, we're recording from rats. We have 128 channels worth of data, and these are electrodes. We're just recording everything 1 hertz up to, I think it's 30 kilohertz. And what that looks like, so here's one channel of data. Here's the rat in a box. I think it was about a 2.4 meter square box. I'll tell you what the red square is later. This is what you see on a typical recording electrode. It's showing you about a second worth of data at the time. So you've got high frequencies, you've got action potentials in there, but you've also got this huge oscillation. By the way, this is known as theta. So if you stick an electrode in a hippocampus of a rat brain, the one thing you'll see before all others is an 8 Hertz oscillation, which is just huge. Humans have it, all mammals have it as far as we can tell, although it sort of slightly varies across different species. And so the question is, if we want to decode quantities about the animal's behavior, things like its position, its head direction, how fast it's going, what should we be looking at in that data? Which bit's informative? Obviously, we could rest on the existing neural theory, or we could just step back and say, look, I'm going to throw this raw data at a network, got 128 channels like that, and I'm going to ask it to decode quantities and see which I can decode and which I can't. And then, in principle, we can look at what the network's learned, turn it on its head, examine the gradients, and see which elements of this code is informative about which quantities. So we tried to do exactly that. So basically, all the pre-processing we did was we did a wavelet transform on this. So we feed each of these 128 channels in as a bunch of different frequency bands with power in them. And we're trying to train a network which at the same time is going to we're training on all of these things at the same time, I should say, by the way, head direction, position, speed. We also tried things like time in the environment, which we could also decode, and various other sort of behavioral characteristics. Again, unsurprisingly maybe, this works really well. So if you think about Euclidean error, so that's the distance between our decoded location and the animal's true location, the network the network's doing spectacularly well. It's down to about a 10 centimeter error, median error, which is about 5%, whereas a sort of standard state-of-the-art in neuroscience anyway, Bayesian decoder is up about 20 centimeters. So we're doing way better than that, but that's not really the point of this. And maybe I should also say it's very efficient. If you start deleting data and subsampling it, then actually it's advantage over the Bayesian decoders. So that's the network. This is how many tetrodes worth of data you've deleted. Tetrodes are just bundles of 4 channels. So pretty much at every level of data it can exceed a Bayesian decoder. But the really interesting thing, and the thing that's promising for the future, is this. We can go back and we can look at the gradients of the network as we're training and ask what's informative. And so some things were fairly predictable. So if you try and decode position, it's these really high frequencies. The really high frequencies are the action potentials. It's just telling you that the action potentials in the hippocampus and entorhinal cortex are informative about where you are. We already knew that. John O'Keefe got a Nobel Prize for it 5 years ago. What's more interesting is this. So we found we could also decode things like speed very efficiently. And speed, actually, you get from the low frequencies, much more so. For things like, as we've now done a bit more analysis, we can see, for example, that theta that I showed you. Theta frequency is modulated by speed. Gamma frequency, which is another frequency band up at about 40, 50 hertz, also very informative about an animal's speed. And so that's starting to tell us things that we didn't know before we started to do this. And I guess that's maybe where I'll leave it, because this is the promising future direction, is that actually we've now got to the point where running these networks bottom up is starting to tell us things we didn't know. And so maybe to very briefly conclude, yes, we can spontaneously recreate biological-like patterns in deep networks. Yes, we can use it to test hypotheses we have about those things, how those things work. And potentially, deep networks are a powerful tool for interrogating neural code and trying to understand what's actually represented. Thank you.
Time for a few quick questions.
Yes, did I interpret your graph correctly that it took tens of millions of training episodes?
Yep, absolutely.
That doesn't seem very
Biologically plausible?
Plausible.
So it's an interesting question. I get asked that question almost every time, actually. It's obviously a very personal question. So it depends what you think each of those steps represent. Each of those steps in time, is analogous to less than a second of time of the agent experiences in the environment. So it's a lot of steps. And indeed, you could learn to do that task in probably 5 minutes. But the point is, when that agent starts training, it can't see, it doesn't know how to move. What we're talking about replicating is not only all the experience you've had in your lifetime since you were a baby as you learn to see and learn to move your body, but also evolutionary timescale. So even before you were born, I knew you were going to have grid cells because you're descended from a long lineage of successful animals that have learned to navigate. So that training is plausible if you think you're basically building your way up from the basic sort of mammalian body plan all the way up to something that can actually navigate. But yes, it is objectively quite a long time.
Isn't that the point that Steve was making about you don't start with a messed up brain?
Well, it depends what you're trying to understand. If you're trying to understand the basic principles of what a grid cell's good for, then you don't want to hard code things in. Like I could already do traditional neuroscience, right, and go and build. Indeed, I have built models of grid cells which are traditionally computational in that I start with my own assumptions about what they might do. We wanted to do something coming from the other end, where it's basically we start with as few assumptions as possible and just say, I think if you have a network that does thing X, then it should do that efficiently if it has this representation. So we wanted to test that. We could cut down the training time if we put in lots of assumptions, but if we put in the assumptions, then it wouldn't really be testing, doing the thing we set out to test.
We've got more questions?
Did you look at, so you looked at speed and direction in the last part there?
Yeah.
Could you associate that with the, whatever it was, 50% of the neurons that you don't know the function of?
Yeah. So that's exactly right. So one of the goals is to understand, all those things you record that you don't understand, which are 50% of the neurons, bunch of the other sort of neural signals, what they're representing. So actually, yes, you can. It turns out that quite a few of those neurons in the entorhinal cortex and elsewhere, which seem to be sort of aspatial, they just fire everywhere, are quite highly modulated by running speed. So they are also informative about it. So yeah, that's exactly the point, is to try and understand what some of those other things are encoding.
I think in the interest of time, we will move it on there. So let's thank Caswell again.